Hi everyone. First of all, thank you very much, sincerely, for inviting me to talk today. It means a lot to me and my team to be able to talk to you about what it is that we're doing as part of our research down at Southampton. So thank you for the invitation, and hopefully the next half hour I'll be able to impart to you what it is that we're doing. Of course, if you have any questions, if anything's not making any sense, just give me a shout, but I'll try and explain as best I can what it is that we do. So I'll start off by talking a little bit about our team. So we are called the Innovation for Translation Research Group. And the reason that we've given ourselves a name rather than just being Zoe's lab or Tim's lab is because I co-lead a team with a surgeon who is an upper GI surgeon. And that's really what got me into working on GIST, because he's been performing these operations for a number of years. He's actually been consenting patients and collecting samples. And we met and realised that my work, which had previously been in paediatric soft tissue sarcomas, actually aligned with some of where he wanted to be able to make use and do some research in the GIST space as well. So he mostly does esophageal adenocarcinoma, so carcinoma that's just at the junction between your stomach and your esophagus, your food pipe, but wanted to do something with these GIST samples. And that's really part of what brought us together, working together, and we named our team this. And we came up with a vision for our team. And for us, this was really important because, as you'll see on the next slide, actually, we do lots of different research. So we're not just doing esophageal adenocarcinoma, and we're not just doing GIST, we're doing lots of different diseases. But actually, what we're trying to achieve does apply across the tumour types and actually some of the rare disease work that we're doing as well. And it helps the team really keep an eye on what it is that we're trying to achieve. Because in biology, it's very easy to go down a rabbit hole of why can't I get my Western blot to work? So that's a scientific it's something we do in the lab, a lab technique, and you can get really into your own head and worried about the little things, but actually we really want our team to focus on the bigger picture at all times and to bring themselves back to why they're doing something, because actually we find that motivates the team, that keeps the team happy, that keeps the team wanting to carry on and pushing through some of the tricky things that you come across in science. So it's really important for us to have a vision. This is just an overview of all the work we do, and most recently we've started working a little bit in GIST. So our focus is really to start to understand response to therapy, so understanding why people respond, why they don't respond, what are the biological, so what are the things that's going on within the tumour and the other cells that interact with the tumour that allows either a patient to respond or not respond. And this isn't limited to chemotherapy, that's kind of where we started, but actually with other therapies now. We want to understand treatment resistance, and we do this across the board. And ultimately we want to find new therapeutic strategies to help with these tumour types. So this is our team. So I'm in the middle there, and Tim is stood next to me, the tall guy. And today we're going to talk about some of the work that these guys have started doing. So this is Jack here, and Jack is really the reason that we actually started doing GIST. So Jack, a number of years ago, he's a PhD student now. He's just finishing his PhD. He's going to be my postdoc, which is super exciting. So we're keeping him in the team. But he actually did the first single-cell RNA sequencing experiment on a GIST sample, and I'll explain what that is in a moment. But that really kick-started our GIST research, which is excellent. And Jack is hugely motivated to continue working in GIST, and he's really led all of the research that we've had funded by GIST Cancer UK and by Sarcoma UK. They've been part of his sort of babies, his projects that he's wanted to do. And so it's great that he's going to stay with the team. And as well as the team, we have some people who are not strictly part of the team, but these guys are integral to the research we're doing, and I'll talk a little bit about what they're doing and how they're integrating with us. So the tumour microenvironment, as Nick mentioned, this is something we're really interested in, so I'm going to talk you through exactly what that is and what that means, and why it's of interest to us and why we think we need to understand it a bit better. This was supposed to animate, but clearly it hasn't. So essentially, the tumour microenvironment is full of not just your tumour cells, but also immune cells and fibroblast cells, so structural supporting cells, and this thing called the extracellular matrix. So your tumour cells don't exist alone. They actually interact with and are supported by some of these other cells of the microenvironment, and your extracellular matrix forms this barrier that kind of keeps your tumour cocooned and encompassed. And what we're starting to understand now is that actually, rather than the tumour cells sort of excluding and ignoring and trying to get on without the support of the immune cells or these other cells in this tumour microenvironment, that the tumour cells actually speak to and even hijack some of these cells to allow them to continue to survive and actually to be hidden from the immune system. And so we think of the tumour microenvironment now in two ways. We kind of have an anti-tumour microenvironment, and this is essentially where actually the tumour isn't necessarily hidden from the microenvironment. And we think there's this state in which, particularly with non-aggressive tumours, and maybe even tumours we don't even know about, where the immune system is able to recognise the cancer and eliminate it the same way it would do with, flu or your chickenpox or anything like that. And so this is this anti-tumour microenvironment, and actually you guys will have heard of immunotherapy, and essentially what immunotherapy does is makes use of this. So it allows the immune system to recognise the cancer and kill those cancer cells the way it would normally kill, your viral cells or viruses and things like that. However, we understand that with a lot of tumours, they actually exist in this tumour-promoting microenvironment, where the cancer cells signal to the immune cells and basically say, you can't see me, I'm not here. And in some cases, they even say, not only can you not see me, I actually want to use you for my own ends. I actually want to make use of some of your properties to help me grow and divide. I want to make new blood vessels so I can get bigger and so that I can move to another part of the body. So we have this tumour-promoting microenvironment where these cancer cells are speaking to these. So T cells, really important part of the immune system. These are the cells that are required to recognise and kill your viruses. And they basically speak to these and say, you can't see me. And then they hijack some of these other ones to basically say, I'm going to grow, I'm going to survive, I'm going to move around the body. And we really want to understand the role of this, particularly for GIST. So we're using a number of techniques that I'm going to go into that are these kind of very new, quite advanced techniques that allow us in a way that we haven't been able to do prior to a few years ago to really understand what cells are in the tumour microenvironment. So some cancers will have these immune cells within the immune microenvironment. Some cancers will exclude them. And we need to understand that, which tumour cells are excluding which immune cells and which ones are interacting with, and how are they doing that, and how can we get the tumour cells to be recognised by the immune system. And we use these techniques. We also want to build better new laboratory models, and for us we're particularly interested in those wild type ones because there are no models really that exist for those. So we're really interested in, if we're going to develop a new treatment, we need something in the lab that we can test it on to see if it's the right thing to do before we're able to progress it into a patient. And this is really important for us because we're really interested in drug repurposing. So really, we want to take a drug that's in use somewhere else and see if we can apply it to GIST, because that means we'll very quickly be able to get into a clinical trial. We don't have to go through all of those lengthy processes to get a brand new drug made and everything else that are very expensive. So if we can repurpose something, then we can get it into a clinical trial quickly, and if it works, get it to patients more quickly. And as I mentioned, this is how we're going to find and test those new strategies with these models. So firstly, single-cell RNA sequencing. So historically, we use something called bulk RNA sequencing. Now, RNA basically are part of the building blocks of what makes a particular cell a cell. So you'll have, you've got hair cells, you've got muscle cells, you've got nerves, you've got all sorts, blood cells, all those skin cells, and they're all different. The DNA in your cell is all the same, but the RNA will be different. So your DNA contains all your building blocks, but in order for a muscle cell to be a muscle cell, a skin cell to be a skin cell, they switch on and switch off different genes. And your genes come from this RNA. So you have more of some RNA and less of other RNA in your skin, more of others in your muscle and less of others, and that kind of thing. And the same is true of a tumour cell. So it'll have different RNA that switched on and different RNA that switched off. And I'll talk a little bit more about it later. But the way we used to measure this was essentially something called bulk RNA. And what that tells you is all of the RNAs in all of your particular tumour sample that are up or down. But what it doesn't tell you is which cells they came from. And as I mentioned from you guys will know you might have had a biopsy or a section. You get a piece of tissue, and actually that piece of tissue won't just contain a tumour cell. It'll contain lots of different cells, but also not every tumour cell is necessarily the same. So some tumour cells might be more aggressive, or they might be a different type more. You might have heard differentiated, more differentiated, less differentiated, and that kind of thing. And the problem with bulk RNA-seq is we can't tell the difference. So my analogy here is like this sort of smoothie here, where you've got a lovely tasting smoothie, but you don't really know what's in there. This single cell RNA sequencing allows us to understand that it was strawberries, blueberries and raspberries that were in our smoothie. So it allows us to see those different cell types. And essentially this is what it looks like. So where we have our bulk RNA, we have an average gene expression, but we don't know which cells it came from or whether there are different types of tumour cells there. Our single cell allows us to say, okay, we've got these tumour cells and that's all the RNAs they've got switched on and off, and this is our immune cell. And the reason for us that that's important is because it allows us to understand if there are, firstly, if there are potential therapeutic targets. So what that means is if there are things that are switched on, genes that's got a lot of expression, so genes that are really high in the tumour cell, that might mean that we could target that with a drug. And the second thing is it does is allows us to understand why the immune cells don't look normal. So, for example, T cells, those immune cells I talked about, we think of them as being either active or exhausted, and when they're exhausted, they don't work. And this single cell allows us to understand, are those T cells switched on, or are they completely exhausted? So this is one of those techniques that allows us to do that. And for GIST, there's actually a little bit of data out there so far. So these are just two things that are that some other groups have put out for the single cell. So a couple of years ago, they published a paper with 2 patients, but both of these were KIT mutant. And then this year, there was another paper, again with the mutant. But this is where we saw a gap. So we're starting to understand for the KIT mutations what these tumour microenvironments look like. And actually we can see, you can see they've got lots of these immune cells in there. But we don't know that for the SDH deficient, for the wild type GIST. So this is really where our work is starting to focus. And this is some data from Jack that he produced, where we can start to see, okay, in one of our SDH deficient samples, actually we also have a lot of T cells. And as we break this down, we start to understand, though, although they're there, they're not on, they're not active. So that helps us understand that although they're present, the tumour has likely signalled to them and said, you're not working. So we're starting to unpick it now a little bit and understand that better. And another really important cell type for us is this thing called a natural killer cell. It's another immune cell, and their job again is to recognise and kill cancer cells. And again, we do see them in GIST, but we're just trying to figure out why they're not functioning. And we think that's because the tumour cells don't have these things called receptors that allow the NK cells to recognise them. So they basically say, you can't see me. You NK cell, you can't see me as a tumour cell, so you don't recognise me as foreign, so you just go off and do something else. And this is really important stuff for us to know. So what's missing from what we've got so far? So the thing that's missing for us, first of all, is positional information. So although we know what cells are in there, we don't know where they are. So although there are T cells in this thing that we sequenced, are the T cells within the tumour cells, or are they being excluded? So where do they sit within that actual 3-dimensional tumour? And the same thing for the stromal cells. Are these things called fibroblasts forming a barrier that means that immune cells can't get in and the cancer's almost cocooned in its own little bubble? So we need to understand not just what cells are in there, but where they are and how they're talking to each other. And we want to understand what those interactions are doing. The other thing is, we don't have any of this information other than the one single cell experiment we've got. about wild type or SDH deficient GIST, because there's nothing really out there in the literature. So this is what we have focused our work on, is understanding where exactly the cells are, and then understanding this tumour microenvironment for the wild type and SDH deficient GISTs. So that brings me to this very clever, very new technique called spatial transcriptomics. And what this does, basically, stick with me. So this is RNA, that RNA expression that I talked about, in those different cell types, but where they're located in a tumour sample. So this is a tumour biopsy, okay? So this is like a slice through a piece of tumour. This is actually a slice through a lymph node. I'll come clean. This is a slice through a lymph node. And what this technology allows us to do is say, okay, what cells have we got? And where are they? And which cells are talking to each other? So for example here, I think 0 is our tumour cells, and actually 6 are some of our fibroblasts, and we can see that they're very close to each other. We didn't know that when we only had the single cell, and now we can start to say, okay, well, are these the green, the T cells? Or, you know, we can start to understand that, so we can start to say, okay, actually, there is infiltration of some of these immune cells, but actually they're clearly not working still. So that gives us that information about what is the problem, because you can imagine if you're not infiltrated, so if your immune cells and your tumour cells are not mixed in together, then that's one problem you might want to solve. However, if they are mixed in, you've got a different problem to solve. So for us, that tells us, okay, they're in there, they're just not recognising the tumour cells, and we can start to understand how we can make them recognise them. So we can start to say, okay, we know they're in there, how do we then get the tumour cells to light up like a Christmas tree to our immune cells? And that's what our lab's are really interested in doing. So the work that was funded by GIST Cancer UK and by Sarcoma UK is basically allowing us to do this. And Dean unfortunately couldn't be here today, but Dean is a computational biologist who I work with. And this is a supercomputer. It's amazing. It's huge. It's one of the biggest supercomputers in the UK and one of the top 500 in the world. It's amazing. So we've got this at Southampton, and it allows us to process ridiculous amounts of data. Like, it's incomprehensible to me. I'm a biologist. I don't understand computers, but it's incredible. So the PhD studentship that is literally about to start next week was funded, as I say, by GIST Cancer UK and Sarcoma UK. And the idea is not only to generate some more of this data, but to build this pipeline that will allow us to do the most we can with the data. So these technologies at the moment, they have some pipelines, but they don't make the most of the data, and Dean is really keen to work with a computer scientist to really see if we can make the most of the data, get more out of it, integrate our single cell and our spatial transcriptomics data as best we can. And then we'll generate an atlas for the GIST. So we'll do this for wild type and for mutant GISTs. And this will allow us this positional information to understand. And we're going to integrate that. And we're hopeful that we will be able to identify potential therapeutic targets, so see if we can find a new way to treat them. And a lot of what we focused on is how we can augment immune therapy. So can we actually use a drug that will allow, as I say, those immune cells to light up, to allow the tumour cells to light up to the immune system, because that will allow anything where you can get your immune system to recognise is preferable to just giving a drug, because then your immune system takes over, that kicks in, and hopefully that creates a memory, so it will stop the tumour coming back. And also we want to look for biomarkers, so we want to use this data to see if we can find reliable biomarkers for clinical use. So the next thing we want to do as well is to generate those lab models, so that when we do find that information from the studentship, we can actually start to test some of what we see. And so this again has just been funded by GIST Cancer UK, where we want to do two things. The next one is quite complicated, but I'll try and explain it. But the first one was to basically use some patient material, so literally some tumour material resected from a patient, to create a model in a dish. And we actually did this a few weeks ago, which is brilliant. So Jack actually did it. So this is from a publication so we've adapted something from a group in the States who've done it. And this is real. So these are our lab models of an SDH deficient GIST line that we're able to now use in the lab. And the thing we also want to do is take the immune cells and see if we can make a model, a three-dimensional, so this is it in 2D, this is it in 3D, that will also integrate some immune cells so we can start to understand about how immune cells do or don't talk to our GIST cancer cells. So this is super exciting, really exciting for Jack, because a lot of people have said, it won't work, you won't be able to do it. So it's amazing, which is why I'm not letting go of Jack. But yeah, the second thing we want to do is do something which Tim, the co-lead, calls the bonkers idea. But essentially, we want to try and use some of these cells called interstitial cells of Kajal, which are thought to be the potential cell of origin for a GIST. So these are these cells that sit in the lumen. They express the same markers as GIST cancer cells, so KIT and DOG. And we want to see if we can isolate those cells because they're sort of stem cell-like. So they're these cells that are able to grow and divide, but they're normal cells. And we think what potentially happens is these normal cells get transformed and that turns them into a cancer. So we want to isolate these cells, and we actually found that although GIST don't have a lot of these cells, a benign cancer called a leiomyoma actually have lots of ICCs. So we're going to isolate them from a leiomyoma, as we have done. So this is our single cell data where we can see them. And we know that, these, they just look like brown dots to you, but that means we know they're expressing KIT and DOG. So we've taken these cells and we've stained them for those key markers, so we know they're ICCs. And then we want to do something called CRISPR. So you may have heard of CRISPR because it was in the news quite a while ago, but it's this gene editing technique that's recently been developed that basically allows us to cut the DNA in a very specific place. And the thing about SDH deficient GIST is they often have some form of a non-functional SDH. And we're going to start with SDHA because it's the most common non-functional subtype. But basically, they have a non-functional copy of this gene. So we're going to basically replicate that. We'll isolate these cells. We'll do this CRISPR knockout and just snip the gene and basically make it non-functional in those cells. And then, so this is just showing that we've done it for another cell type. So again, Jack, absolute wizard, has managed. So this basically shows that in a wild type cell, in a wild type stem cell, you have lots of the presence of that particular gene. And then when you do this CRISPR, it's gone. So that tells us that the techniques worked. And then we'll hopefully monitor how they develop and see if they start to look more and more like a GIST. We'll see. We'll see. We've also got another few different things we're going to try. But I thought I'd finish lastly with what else we want to do next. So I am a collector of people, and I feel so passionately about sarcoma that I like to encourage everyone around me to also feel passionately about doing sarcoma research. So I'm busy making just about everybody I come into contact with at Southampton want to work in GIST as well. So the first thing we want to do is have a look at other ways that we might implement some of the really cool science that's happening at Southampton. So some of the stuff that other people are doing, not my team, to see if we can find other ways that we might be able to treat and think about GIST. So the first thing is, this is a guy called Matthew Blunt. He previously worked in leukemia, and he's a big leukemia research fellow. Very, very, very good guy, very clever. And he works on these natural killer cells that I told you about. And essentially what happens in a normal situation with a cancer is the cancer cell is hiding from these NKs, as I said. So the NK cell just can't see the cancer and it just goes off and does its own thing. But Matt's lab is working on developing something called a CAR-NK. And to cut a long story short, essentially this is an engineered NK cell that is taught to recognize the cancer cell. So you remove this lack of recognition, and the NK cell says, I can see you and I can eliminate you. And this is what Matt's been doing in other cancers, as I say. You might have heard of CAR T cells, but there's a big advantage of CAR NK cells over CAR Ts, because the problem that they're seeing in clinical trials with CAR T cells is that they have, they're something called allogeneic, and that means that you get graft versus host disease, which is very, very nasty, and means that actually they might reject the CAR T cells, so it doesn't work. You also have some cytotoxicity, so this is something called cytokine release syndrome, and this is immune effector cell-associated neurotoxicity syndrome. Nailed it. Nailed it. And so again, the beauty of this new engineered NK cell method is that you don't get any of these cytotoxicities associated with the use of this that you see in T cells. And the really important thing is, these things are, CAR Ts are already in clinical trials. And so again, these things are starting to enter clinical trials. And Matt's done a little bit of preliminary work and found that he thinks there are things on GIST cells that we might be able to use to engineer an NK cell to recognise it. So really promising. So we put in a little application into Sarcoma UK, just gone, so fingers crossed. And then the second thing I wanted to talk about is this is another guy called Matthew Rosarilli, and again he's worked in blood cancer and some esophageal adenocarcinoma. And he's interested in another cell process. So as I've sort of talked about already, your DNA gets transcribed into this RNA, which eventually becomes your protein. And again, as I mentioned before, the difference is in your normal cell, you have certain genes that are switched on and certain ones that are switched off. And in a cancer cell, they're different. And what we're starting to understand now is this very complicated process. But this is a piece of RNA, so trust me, this is a piece of RNA. And at the end of your RNA is this thing called a tail. And when in a normal cell, you have normal tail length. But now we're starting to understand in cancer cells that actually they have the wrong tail length, and it can change the way your RNA looks. And that in turn changes whether it's on or off, but in the wrong place. So we think that that's what basically says your cancer cell has got these tails that are all wrong. And essentially what we think is that actually, as I say, with the tails, in your cancer you have very aggressive cancers that are poorly differentiated. They grow and divide a lot, but actually they have these very, very short tails, and that leads to very, very high levels of RNA that should be low. So for example, things that are oncogenic, so things are called oncogenes, that basically are the genes that keep cancers growing and divided that should be switched off are on. And then again, tumor suppressor genes, so these are genes that we know are involved in basically suppressing the formation of tumors. They have much longer tails and are switched off. And actually now we know that we can actually we can target this. And again, Matt's found some really, really interesting preliminary data in our GIST data to suggest that this exact process is happening in GISTs, and it's a process that changes as people become resistant to imatinib, and also it's different in SDH deficient GISTs as well. And again, so we're hoping that he's put in a studentship, I think, to Sarcoma UK, but one way or another, these guys will probably do this work because they feel really passionately about it, because I make them. And that's really all I wanted to say, other than to say, to really thank the people who do this work, because I get to stand here and talk about it, but actually, people like Jack, and this is Dean here. Zahor is a computational scientist who's working with us on the PhD. This is Ben Sharp, who's also done some of the spatial for us. These are the guys who really do the work and who make everything that I stand up here and talk about possible. And finally, I just want to thank you guys so much for giving me the opportunity to come here and talk to you about what it is that I do. So thank you very much.
Tony, how long have we got for questions? So a couple of things. Okay, for questions. Is Robin here? Oh, yeah, good. So a couple of things. I applaud your ability to bring this down to a level that even I understand. We've had, right when I first started doing this, it was so complicated. I'm sat there thinking, I have no idea what they're talking about. But thank you. That was really good. Really, really good. And it just also shows the collaboration element. You talked about America. You talked about other research institutions. This collaboration really does make a difference. And just to let you know, the way that we decide whether or not to offer grant funding to people like Zoe and the projects that she's coming to us, we don't just sit around the table, our trustees, and say, yeah, that looks good, we'll go with that. We put it to what we call our medical advisory board, which is a group of eminent GIST specialists who know as much as there is to know about GIST, but are always learning. They look at it in detail. Once they've decided that, yes, they think this looks promising, we then send it to peer review to America. I don't know if you know this.
I don't.
No, right, okay. We then send this for peer review in America. So we ask the American specialist to also look at it and tell us whether or not they think what is being asked for and the amount and what they're going to do and the potential outcomes are worth us funding. We reject some in that process and we support some in that process. So it's complicated, and I chair, along with Tony and Jane, some of those medical advisory boards, and we sort of sit back and listen to them talking to each other, and we have no real clue what they're talking about, but clearly they know what they're doing and what they're saying. So that's just to let you know that it's done properly. So any fundraising that you do that we put into that side of it, it's done properly. So we've got how long for some questions now? 5 minutes. So do we have any questions?
Hi, I'm Rachel. I think I heard you say that the single cell RNA sequencing could lead to a tumour marker for GIST, is that right?
That's what we're hoping that we'll be able to use this data for, that we'll have enough data from enough different types that we'll be able to understand where there's something that effectively correlates with something. So for us, we tend to say, okay, what are all the markers? What are the tumour markers, the immune markers? And what correlates with, say, a poorer outcome or the recurrence of disease or metastasis or some clinical thing? And we're hoping that we can derive those biomarkers through the data that we're generating. So yes, essentially.
That's very exciting. Yeah.
Thank you.
Hello, my name's John. Nice to meet you. I hear from time to time of people spontaneously recovering from a cancer. And does that give a lead into research into your work?
Yeah,
I understand it happens maybe after an inflammation or something like that.
Yeah, so one of the things that we do in our team is we like if we can get an early biopsy or an early sample, and if we understand the journey that happened to that patient, we can ask those questions. So if we know that we've got a sample and we understand what that sample looked like when it first existed, and then we know what happened to the patient, if they spontaneously regressed, we can exactly go in and ask that question. What's different there? What's different about that patient, that tumour microenvironment, and get exactly that, an idea of, is there something different? Is there something different, particularly for the tumour microenvironment? So you mentioned inflammation, that is exactly key to understanding that, and if we've got those samples, we can absolutely do that sort of thing, yes.
Oh, making me work today.
Thank you so much, Zoe. I agree, that was very accessible and really interesting. I'm curious, does the tumor microenvironment change if it's a primary versus a secondary location for the cancer?
Yes, it absolutely does. So there's another paper where they show it in the KIT mutant, but they also look at something that is either a metastatic one or a primary one. So there's the met and the primary, what we call it. There's also the recurrent, and you might have a local recurrent sample, and again, those are different as well. So the work in our team, we've started doing it in the esophageal adenocarcinoma, but we want to also do it for our sarcomas, including GIST, is ask that question of, if we've got a primary, so for one of our patient samples, we've got 6 different locations, and we can do exactly that and say, what does it look like in what we think is the primary, and what does it look like in these other sites? What's different? And also then ask that question about the lymph node, because we now know that those lymph nodes that are part of your lymphatic system, part of your immune system, that they play a really key role in the ability of a tumour to carry on growing and dividing and to move around the body, and we can do exactly that. But again, what happens in a lymph node and what happens in a primary tumour are very different, but these techniques allow us to do that and understand those differences. Yeah.
Right, one more question.
Hi Zoe, I was fascinated with the tail of the cancer tails, and I just thought, I've never seen or heard anything that was really new. So that leads to a build-up of RNA. And then what happens? What did you
you get more protein, and those proteins are, the proteins are the real building blocks. So we look at the RNA as a kind of surrogate because it's much easier to look at than these proteins. But lots and lots of proteins means you've got something different going on. So there's a particular protein that we know can be targeted with a drug, and it basically drives lots of expression of these genes that are pro-tumour and switches off this expression of things that are anti-tumour. And so that's one of the things we think is going on with that. So you have this different tail length, lots of a protein. It then signals to other things and says, I'm going to make a very growth permissive environment so my cancer cells can keep growing and keep dividing and be aggressive and that kind of thing.
Fantastic.
Thank you.
Right, thank you. Oh, can you hear me? Yeah, thank you. Zoe, thank you very much.